Hello everybody, this is lesson 4 in the anatomy muscular system unit. So today we're going to take a look at some of the microscopic, or even more than microscopic, the chemical structures inside the muscle. So this is a real deep dive. So we talked about the two most important proteins that are associated with muscles last time. We talked about actin and myosin. So we're going to look at how these two proteins really work together to help make this muscle work. So we'll start off with actin. Actin is actually the simpler of the two, and it's a very, very common protein. And right down here, it's shown as the purple strand, and as you can see, it's sort of a twisted string of pearls is sometimes what it's described as, and it's often described as a thin filament because it is thinner than the myosin. There are some, now we talk about actin and myosin, and they're certainly by far the most important, but there's some other accessory molecules that we're going to talk about as well. So one of them is this molecule called troponin. Troponin is this little red molecule right here, and it hangs out on the side of the actin, and its job is to provide the calcium binding sites. So we briefly mentioned this last time, that the calcium is actually what triggers the muscle to actually start to contract. So in order for the calcium to work, the calcium actually has to have a place on the muscle, on the muscular system unit, that actually will allow it to bind and create something that happens that makes this go. And it's a little complicated right now, but we'll get into it. But this molecule is where that calcium binds to. And then additionally, there are these really thin strands called tropomyosin. Tropomyosin are really, their job is to prevent muscles from contracting. So if without the tropomyosin, you would actually have a muscle that would grab onto each other and just cling, and it wouldn't actually stretch back out, it wouldn't relax. So this actually blocks these little yellow sites right here, are called these binding sites, and what the tropomyosin does is blocks those binding sites, which means that you can't contract the muscle. So what we have to do in order for the muscle to contract is we actually have to move the tropomyosin. So the essential cascade of events that happens is that when your body is telling the muscle to move, you release calcium, and that's stored in the sarcoplasmic reticulum. That's that specialized endoplasmic reticulum inside the cell. That calcium comes, it binds to the troponin. The troponin then moves the tropomyosin and then frees up these little yellow sites, these binding sites that allow myosin to attach. We will have videos, don't worry. And then we have the myosin myofilaments. They are considered thick because they are considerably thicker than the actin. Some people describe them as mini golf clubs. There's all kinds of different descriptions out there. But the key part of the myosin, this part is just this long, thick part here, is just really strength, and it's to provide an ability to, pull on each other, because this molecule actually has two ends. It has one end that faces one direction, and then the other end looks like a mirror image of this. And so you actually have these sort of heads at both ends, and they're facing opposite directions. So the head is the part that actually can bind to the actin on those little yellow dots. And that's what's going to happen when we actually start contracting the muscle. So sarcomeres are the basic functional unit. So we're going to take a deep dive into what is a sarcomere, how does it work, and what is going on when the muscle actually contracts. They are connected end to end, sarcomeres are, and each neighboring one is going to contract, and each one by itself can only contract a very, very short distance, but collectively they can considerably shorten a muscle. So this is a sarcomere, and I'm going to move myself up here. So the functional unit, this sarcomere, goes from what is called a Z disc, and these are the Z discs right here. So from one Z disc to the next is considered a sarcomere. Just so you know, the Z disc can also be called a Z line. They're interchangeable. I've seen them both ways, even within the same textbook. So the Z discs are really the connection point between one sarcomere and the neighboring sarcomere. But what we're going to look at is what's inside that sarcomere, what's going on inside there. And what the Z discs are an attachment site for the actin. So the actin is anchored here. And notice the Z disc anchors one sarcomere actin right there, and then it also anchors the neighboring one right there. And so you actually have this ability to connect those two sarcomeres together. And that's critical, of course, because when you want to shorten the whole thing. So what we're going to see that when this muscle actually contracts, that this Z disc and this Z disc are actually going to move towards each other. And they're going to shorten, and they're going to go towards what's called the M line, which stands for the middle. Okay? And what we see here when we look at this diagram is that we see a bunch of bands. And this is when we first looked at the muscles under a microscope, we could actually see light and dark bands. And that's where these names come from. It does get a little confusing, I understand, with all the different labels, but we're going to work through it and you're going to start to see it. So what we see is that we have what's known as the I band. Okay, I band is actually relatively, it's dark, but not the darkest, okay? And the I band is where we actually only have actin. And so it's actually, I should say that that's actually the lightest of the colors, the I band, because actin is the thin filament. So this area right here, and the Z discs is right in the middle of that I band, okay? And then there will be another I band over here, okay? They just sort of repeat over and over. The A band is from one end of myosin to the other, and that's darker. Now, obviously, this area right here is even darker because it's got actin and myosin. But the A band is generally darker than the rest because myosin is thick and it blocks more light. You then also have this thing called an H zone, where it's only the myosin, no actin, okay? And the M line is right in the middle of that. That's the middle. And then you have the I band, A band, I band, A band is the pattern you see when you look at this muscle under a microscope. And what we were able to see actually is we were able to make muscles contract under a microscope. And so what we could see is what happens to these light and dark colored bands as the muscle contracts. And that's it. We'll get into the actual muscle contraction in the next notes. I understand this gets a little confusing and you may have to go back multiple times here. Terminology is tough. This is a really tough subject, so please keep with it and ask lots of questions and use all the resources we offer you to help you out here. All right, thanks.